welcome back to the lecture series on design for biosecurity. So, we'll be starting this week with a bit of a recap of what all we have covered, and then I will go to the last case study, which is insulin. So, if we look back where we started this journey, so one of the fundamental things what I highlighted in the beginning is the need for economic sensors to meet the emerging threats of biosecurity. This was since second week we have been exploring. In that process, we talked about the filovirus causing Ebola and Marburg sensors, case study for Bacillus anthracis, and eventually anthrax sensors, case study for botulism toxins, and the sensors associated with botulinum toxins. This is where we concluded the last week. And today we will be starting with case study of insulin. Before I get into the insulin story, so this is how we traveled our journey with the Ebola. As I mentioned, please go through this book by Richard Preston about the hot zone. And we talked about the virus, then how it was identified, and the epidemic across, then the mode of transmission by zoonotic pathogen, then the routes by which it entered inside the human body, and what are the places where this virus survives, then the structure of the genome, then how it affects the hemorrhagic fever and all other problems. Then we talked about the different kind of sensors which are being used. Then we talked about the countries where the epidemic has spread. Thereafter, we talked about anthrax. When we talked about the anthrax, we started with September 2001 anthrax attack and how anthrax remains in the ecosystem and get transmitted. Then what are the different types of anthrax, then the symptoms of anthrax, especially cutaneous, inhalation, and gastrointestinal, and about anthrax prevention, and especially anthrax at certain jobs, and the possible routes of transmission, the global map and local insight, anthrax map of 3 continents, why anthrax is a potent bioterrorism agent, and that's where we talked about how anthrax has been used as a bioterrorist agent, and all the gory details of one such attempt which happened in last 25 years, then how FBI got involved into it. Then we talked about the anthrax sensors from here onward, and then we Sorbre equation, quartz crystal microbalance. Then we talked about the different configuration of QCM. Thereafter, we moved on to the theory of QCM and then coupling QCM with electrochemical setup. Thereafter, we move on to AFM, atomic force microscopy, how it is being used for different forms of sensors, and we went into further details about contact mode, non-contact mode, intermittent mode of AFM. Then we talked about infrared scattering, scanning, near-field optical microscopy. Then we talked about near-field Raman imaging, then tapping AFM IR. Then we talked about Raman and IR spectroscopy, a bit of recap, and then we talked about the Raman shift and difference between Raman spectroscopy and IR spectroscopy. And this is one of the chart what I showed you about what are the moieties which are being identified in this process, the complementary relationship of Raman and IR, then the application of Raman, Raman imaging. Then we talked about the laser selection for Raman spectroscopy for development of sensors, then the challenges of removing fluorescence from Raman spectra. Then we talked about AFM cum electrochemistry workstations, which is kind of slowly getting miniaturized versions, and this is something which is being liked across the world. Then we talked about the recognition element of monoclonal antibodies, peptides, aptamer, epitope, and phage display, monoclonal versus polyclonal antibodies. Then we talked about the production of monoclonal antibodies via hybridoma technique, via fast display technique. These are the two major techniques we talked about. Then we talked about the next element of the smaller elements of the antibodies, single chain variable domain. Then we talked about aptamers, then the phage display. Then we talked about surface plasmon resonance and how surface plasmon resonance is a label-free system which is being intensely getting huge amount of user friendliness, and there are a lot of innovations which are happening. And then we talked about how SPR events are being measured and the shift due to the binding on the top of the chip. Thereafter, we went on about design and fabrication of lateral flow immunoassays, LFIAs, and we talked about all the microfluidics aspect of LFIAs and how they function. Then the competitive assays, followed by sandwich non-competitive assays, the positive control, negative control, and all other different paraphernalia involved with it. Thereafter, we move to botulinum toxin. So, we talked about how the transmission of botulinum toxin happens, the case study of botulinum toxin and the key points, and we talked about the overview, then the symptoms of foodborne botulism, exposure and transmission, foodborne botulism, infant botulism, wound botulism, inhalation botulism. And whenever we talk about anything like inhalation botulism, that means this is a potent bioterrorist agent other types of intoxication. Then we talked about a very interesting aspect of Botox, that is the Botox therapy, how this agent could be used for fashion and beauty industry. Then different types of Botox injections which are being used, type A, type B. Then the benefits of Botox therapy, then the crystal structure of botulinum toxin, the mechanism of actions, especially at the synaptic zone where botulinum toxin enters and attack the SNAP25 structure and chopped it off, and how such SNAP structure chopped part are being used for developing biosensors, especially electrochemical SPR based immunoassay for multi-analyte detection. This is the electrochemical assay where you have methylene blue is acting as a electroactive agent, and by chopping it off, how the electrical signal falls down. Then we talked about SPR assays which are being used for botulinum toxin detection, and we talked about the different parts where this acts, botulinum toxin acts on SNAP25. Then we talked about how the SPR detection assays occurs, and this is how the signal is being detected. Then we talked about immunoassay chip for multiple infections. Say for example, if you have botulinum toxin and staphylococcal enterotoxin B in food, how we do the detection assays, and this is somewhere in the last class we wind up about different kind of food sources and where all botulinum toxin is present, and the need for multi-analyte detection. So, this is where we concluded the last class. So, you realize through this journey we have picked up different kind of microbial aspects, starting with the virus, then bacteria and their spores and how they all function. But now this class and in next 3-4 classes, we will talk about a compound which has a potential to murder somebody. You can use it to kill a person. Is a molecule which is produced by our own body called insulin. You all have heard about that there is an increasing challenge among Indian population of diabetes, As a matter of fact, India is considered as the diabetic capital of the world. There is almost an epidemic proportion of people suffering from diabetes in India. Now, there are different theories why it is increasing, from lifestyle to food habits. There are several aspects to it. But the fact is, we are at a crossroad where diabetes is a challenge. And when we talk about diabetes, there are a couple of things, even as a layperson, will come in your mind. One is, you must all have heard about insulin therapy. Those who suffer from diabetes goes through insulin therapy. And the another word which comes, oh, you should not take more sugar, your sugar metabolism is being compromised, and likewise. So what we will do in next few classes is demystify the process. Considering that you all have come from different backgrounds, we will try to demystify the story of insulin. And insulin is almost now 150 years old story. And story of insulin is filled with different kind of controversies. It's really a very amazing thing. First, its discovery, followed by the zone where it was used for as a therapeutic agent against diabetes. Those scientists and their life and the controversies which erupted even before they were awarded the highest honor humanity has developed for scientists, the Nobel Prize, followed by how the technology was taken over by one of the pharmaceutical giants, Eli Lilly, and since then how human life has improved to the point of today's economy, where once again with the increasing cases of insulin, how monthly expenses or medical cost has influenced the life of people who suffers from diabetes. Then there are different kind of diabetes, type 1, type 2, and what are those? And in between, since 1950 to 2000, even now also it's going on, the controversial and extremely controversial cases where people have been murdered using insulin. So it means there are cases where insulin has been used as a murder weapon. So from this point, for next few classes, we will try to demystify the story of insulin. So in the slide, what you see, there is a picture. It's a model which was made, and what is written is, from all who owe their lives to insulin, with heartfelt gratitude. The gentleman whose image you see here is John MacLeod. He was one of the forgotten heroes of medical science. Who was involved in what we today call as insulin therapy against diabetes. So if you look at the story of insulin, the key part or the breakthrough consisted of the work of Frederick Banting, Charles Best, James Collip, and John MacLeod. These four Toronto-based researchers discovered and purified insulin, creating a new effective treatment for diabetes. But this was not that simple a story. And today, I will start to share the story of these four people. So if you look at it, the early 1920s, Frederick Banting and Charles Best discovered insulin under the directorship of John MacLeod at University of Toronto. With the help of genes called insulin was purified, making it available for successful treatment of diabetes. Banting and MacLeod earned the Nobel Prize for their work in the year 1923. But was it that simple? Is what we are going to discuss. Now, at the turn of 20th century, minded this is we are talking about 1900, a strict low calorie, no carbohydrate diet was the only effective treatment for diabetes. So, that means if your body cannot take care of carbohydrate, then you do not take carbohydrate. That was only therapy which was there. So, that means abstinence. You can't handle it, you do not take it, which is fine, but the point is your brain can only use simple glucose sugar for its functioning. So, if you do not take sugar, so if you have a craving for food, so you have to take sugar or else you will faint. So, whenever you see people are suffering, they are giving glucose water. What is glucose? It's basically enough energy for your whole body to absorb, because simple sugars are the one which our body absorbs very fast. That all is a very simplest way, but then if you go into the history, there will be a lot of different aspects which will come forward, but we are not talking about that. But at this point, glucose is the molecule which is absorbed by the brain and our body satisfies its need using glucose. Now, if you refrain from carbohydrate, you go for a low carbohydrate or low calorie food. But this method with food intake sometimes as low as 500 calories per day had its consequences, as slow starvation like diabetes drained patient of their strength and energy, leaving them semi-invalid. The diet treatment also required an inordinate amount of willpower, because you are refraining from taking sugar or something sweet on part of the patient, and very few of whom were able to maintain low calorie diet over long term. So, this is not a, what I call as sustainable therapy. So, it is a non-sustainable therapy. In 1921, researchers from University of Toronto began a series of experiments that would ultimately lead to the isolation, commercial production of insulin, a pancreatic hormone essential for metabolizing carbohydrate, and the successful treatment of diabetes. This is in nutshell, but then what the story was. This is where we are going to now devote our. So, setting the stage for discovery of insulin, and that is where we are going to start our next class, where we will talk about how the discovery actually happened prior to 1900, what all humanity knew, and from there with those bits and pieces of information, how we proceeded to the commercial production of insulin and saved humanity from one of the very critically challenging problem of diabetes. Thank you.